The topic of this video is how we can image electrical activity in the brain. Now, in the last lecture, we talked about electrical ways of monitoring electrical activity in the brain. Now, that kind of makes sense because neurons are intrinsically electrical. So how do we do this with imaging and why we want to do so? That's what we're going to get into in this video. So I wanted to remind you about neural transmission and the fact that in the synapse video, we talked about one particular part of the synapse, which is that there are these voltage-gated calcium channels, and that the action potential, once it arrives in the axon, causes an influx of calcium. This is the key insight here because this is going to be the basis of how we're going to turn electrical activity into optical activity that we can image with a microscope. These calcium is tightly regulated because there's a transient influx of calcium into the cell every time there's an action potential. And calcium is tightly regulated because it is used as a second messenger, and so the cell really wants to actively get rid of the concentration of calcium every time after the action potential happens so that they can happen again and use that as a message for the second time. So what we're going to do is we're going to take advantage of this observation that when there's an action potential, there's also voltage-gated calcium channels that are open, and calcium rushes into the cell, but it very quickly disappears. So knowing this observation, what we can do is make the approximation that we can record neural activity through fluorescence calcium imaging. So instead of measuring electrical potentials through electrodes, we're instead going to image the intracellular concentration of calcium. So how do we do this? How do we turn calcium concentration inside the cell into electrical, which is a proxy for electrical depolarization, into photons that we can image from a microscope? There's two categories of techniques that are popularly used. People have been developing these for many decades. The first category is what's called calcium-sensitive dyes. There's a popular one here called Fira-2. This is a very popular one that have been used, and it is a calcium-sensitive dye that is a small organic molecule, medium-sized organic molecule of this kind. You can buy it in a little vial. It's not cheap, but you can buy it. That's synthesized from chemical companies like Sigma Aldrich. And what you can do is take this calcium-sensitive dye, and if you apply it to a piece of neural tissue or even neurons in isolation in a dish, and let it soak in for a little while, right? Like after half an hour or so, the neurons are going to have soaked in these calcium-sensitive dyes, and the calcium-sensitive dyes have the property that when there's calcium around, it fluoresces in a way that you can measure under a microscope. This is super cool, right? This is cool. However, there's still a problem, which is that you can only use them for a limited period of time. Sometimes they're kind of toxic to the cells. Okay, so calcium dyes, they're a little finicky. Sometimes after a couple of hours, they just don't work anymore. They've bleached. They're kind of toxic to the cells. So the cells start being not very happy afterwards. And you have the fundamental problem that you have to somehow get them into the cell somehow. And this is either an injection, or if you isolate a neuron, it's a little bit easier because you just kind of like pipette it and you can just put it in the bath and it's totally fine. But if you want to measure something that is inside the brain when the animal is intact and actually doing something, then applying calcium-sensitive dyes is a little bit challenging because it's hard to get it in there and then get the animal to be happy and surviving afterwards. So a different strategy is, instead of having calcium-sensitive dyes, we have a class of proteins called calcium-sensitive proteins. And these are engineered proteins. They're not native proteins that are found in nature, but they're engineered by rational design using parts we found from nature in order to engineer something that is a protein that is coded by a genetic sequence that we can insert through genetic engineering into different types of cells, and then have the proteins express inside neurons where you want them to. And like the calcium-sensitive dyes, these proteins have the property that when they're bound to calcium, when there's calcium concentration inside the cell, they fluoresce. And they can fluoresce in a way that is proportional to the concentration of calcium. So there's more calcium, they're brighter. When there's no calcium, they're dark. So that's the property of both of these types of molecules, the calcium-sensitive dyes and the calcium-sensitive proteins. They're either brighter when there's calcium or they're darker when there's calcium. In any case, there's a difference in their fluorescence when there's calcium. So one more thing about these calcium-sensitive proteins that are really cool. I'll say this a couple of different times, just let it sink in, okay? The fact that they're proteins means that we know the amino acid sequence, and it also means that we can design a gene that we can insert into the animal cells so that they express this protein. If we knew the promoter for some proteins that are only expressed in certain types of cells, so for example, I only want my calcium-sensitive proteins, these proteins to be expressed in neurons, so I get a neuron promoter, So other cells in the body don't express this protein. If I wanted to be expressed in only dopamine cells, I would find a dopamine promoter that only dopaminergic cells express. I glue this protein to my dopamine promoter, and now only the dopamine promoters will express this calcium-sensitive protein. This allows me to engineer the delivery mechanisms so that I can measure the electrical activity, the neural activity of only a subset of the populations of cells in a very precise cell-specific way. I will point out that this is something that is categorically impossible to do with electrical recordings because you never get that specificity. You get whatever cells are talking, you don't get to know what they are. Here we have something that we can do that we could not possibly do before with electrical recordings. Now, these calcium-sensitive proteins I told you were made by rational design in the laboratory. And so I'm going to talk a little bit about how that works a little bit because I think it's a really fascinating story. So this type of, there's a couple of different ones. The most popular one that basically everyone is using now, it's called GCAMP. I'm going to talk about how GCAMP is made. So GCAMP is made by, it's a Franken protein that was glued together from a couple of other proteins and made to work, okay? So GCAMP has one part that is a GFP, a green fluorescent protein. Green fluorescent protein is a fluorescent protein that was isolated from the jellyfish. Okay, so this is the same GFP if you've heard about it in molecular cell biology in a different class, same GFP here. But what they've done here with GCAMP is that they've taken the GFP and they've glued it to two other molecules. One of them is a calmodulin, which is a small protein that binds the calcium, and it's stabilized by an M13 protein, which is a part of the myosin light chain. Now, this exact Franken combination does not exist in nature. But all of the parts, the GFP, the calmodulin, the M13, do exist in nature, and so they're kind of glued together in a rational way so that in the absence of calcium, the GFP is in a state where it's not functional, and if and only if, when calmodulin and M13 bind to 4 molecules of calcium, does the whole GFP barrel close, causing it to function as a regular GFP. So in other case, we have a GFP that is now gated by calcium. Only in the presence of calcium binding to calmodulin and M13 do we have a functional GFP. Okay? That's how it works. So GFP part is, of course, the part that's responsible for fluorescence. I'm just going to remind you a little bit about how GFP works. GFP is a green fluorescent molecule. And the way it works, the way we can take advantage of it in these experimental systems is that it has what's called an excitation spectrum and an emission spectrum. I've drawn the cartoon version of it here where we're talking about the wavelength of neurons, right? So longer wavelength neurons are more red, shorter wavelength neurons are more blue. And so the excitation spectrum is where you have to zap the GFP, where you have to hit it with some neurons, with photons of that wavelength, and then you measure at the peak of the excitation peak to see if the GFP is emitting photons. Okay? Same exact thing here with GCAMP, except that this emission and excitation spectra is dependent on the presence or the absence of calcium. And so the same exact thing here, except that you zap at whatever the excitation spectrum is, and you measure at whatever the emission spectrum is supposed to be. In the presence of calcium, you'll see photons emitted at that wavelength. Without calcium, you won't see anything at all. That's the trick. That's the entire trick. With calcium, the GCAMP emits photons. Without calcium, it does not. Now, I told you that people have been engineering GCAMP for a long time. There's now, I think, up to 7 different iterative generations of GCAMP that keep getting better and brighter and faster. There's also different versions of GCAMP that are made with different colors, so you can actually have multiple ones in the same experiment, right? So if you have one set of GCAMP and one set subpopulation of neurons, let's say the glutamate neurons, they emit in green, and then you have another population that is the GABA neurons, and they emit in far infrared or something. You can actually simultaneously image the neural activity of different subpopulations of neurons in the same experiment by having them be separated by different types of GCAMP so that they can be separated in spectral space as well. So super cool technology. Now, GCAMP, what it allows you to do is see if there's fast enough individual action potentials. I'm going to tell you an example what that looks like here. On the horizontal axis is time, so time going to the right. Okay, and what you'll see is this is basically a recording of what's going on in the cell. So every time you see a big spike here, this is an individual spike. So you hit one spike here, one spike here, 2 spikes here. They're really close together. And what you'll see is every time there's an action potential, because there's an influx of calcium that's associated with that action potential, there's there is a corresponding increase in the fluorescence of the G-camp at exactly the same time with a short time delay following that action potential. When there's one action potential, it goes up a little bit. When there's two, it goes up more, okay? Here you see that there's 3 action potentials, one a little pause, one a little pause, another one a little pause, so it goes up in 3 little steps. Here the cell gets really excited and it fires 6 action potentials in rapid succession. So you'll see that this is what's causing this giant, huge leap here in the fluorescence of the G-camp signal. So the fluorescence is much, much higher when the cell is firing 6 action potentials at the very far end of this diagram on the other side. A couple of other things I'll point out about this diagram. You'll see here that the individual action potentials are super short. At the resolution of the screen, you actually don't even see the time course. You don't see that characteristic shape of the action potential. All you see is a single spike. Okay? The same is not true of G-camp, because the G-camp, because of its properties, because the calcium actually takes a little bit of time to get cleared out of the cell, and then the G-camp, the protein itself, takes a little bit of time going back to the conformation where it's not excitable, where it's not emitting light. So instead of going down as quickly as the electrical potential does, the G-camp signal, you can see it goes down exponentially, but much more slowly than the electrical potential. So there's an after effect of the G-camp because of the G-camp itself, because the measurement makes it go down more slowly, not because the cell is actually electrically active at that moment. That's a key point, okay? The second thing that I'll point out that's really interesting about this type of system is that the effects do not actually become proportional to the actual fluorescence. If you pay attention to the axes there, you'll have, those of you who may be astute have noticed that we're not actually plotting the fluorescence of G-camp. Big F is fluorescence. So we're measuring fluorescence, we're measuring through a microscope the photons that are coming out of the cell. But that's actually not proportional to the electrical activity. What people usually do is turn that into what's called a delta F over F. We're looking at the change in fluorescence in a given unit in time. This is the derivative of fluorescence. And then we normalize that by fluorescence. In the previous time step, the details are, there's a lot of different ways to do it. So I'm skipping over the details because it's not totally well established exactly how to do this. People do it slightly differently. But the philosophy is the same. You're looking at the change in fluorescence instead of the actual number of photons emitted by G-camp. And that is the quantity that is proportional to the electrical activity of that cell. These are 2 important points to know about G-camp recording. So, all right, one more thing. If this point doesn't make sense to you, don't worry about it. It's kind of a quantitative point. If you look at the way that the G-camp signal, this delta F over F signal, decays after every action potential, it does have a characteristic time scale and a characteristic shape. What it means is that that shape is if you know that filter, you can basically characterize it as a convolutional filter. So the G-camp signal, delta F over F in time, is the product of a convolution filter applied to my action potentials in time. So if I can apply a convolutional filter and characterize a convolutional filter to the timing of the action potentials, I can also deconvolve the G-camp signal to then estimate the underlying electrical potential. And it's in that way that I can decompose something like this, right? This really big one here. I don't see 6 individual times the G-camp goes up. It never went up 6 individual times, but it went up by that much, okay? So by applying the deconvolution, I can back out that there probably was 6 action potentials that led to that very large increase in delta F over F at that part of the experiment. So those are kind of like really cool details that are in the technical weeds of doing this type of calcium recordings. So I told you that G-camp has gone through, it's super successful, lots and lots of people use it, and there's also a ton of different labs that are actively developing this technology because it's really useful. What's been done in recent years, and in picking the right G-camp to use, a different version of it to use, is you're thinking about the sensitivity of binding to calcium. You might want something that is very sensitive and binds to calcium easily, so it's a more sensitive measure of calcium influx. You might be paying attention to this excitation and emission spectra. I told you about earlier, there's different versions that we can pick. So there's not GFP, but there's YFP and red fluorescence protein, different versions of GFP that can be glued to calmodulin to yield G-camps of different colors. So you can pick your color. You might want to pick your brightness, right? If you want something super fast, you might want it to be really bright. If you want it to be kind of slow, then you can have something, you can get away with something that's more sensitive, but less bright. Okay, so these are all trade-offs here. There's no one perfect GCamp version that does excellently at every single one of these different criteria. And the fourth criteria is the kinetics of fluorescence intensity change, which is that convolution filter I talked about, right? So the convolution filter makes it so that every time there's an active potential, GCamp signal delta F of F goes up, and then it comes back down. Exactly how quickly it comes back down, does it come back down really, really slowly, or does it come down pretty quickly? Those are things that are engineered properties of different versions of GCamp. And so depending on how quickly you want to be seeing changes in that particular neuron population you're imaging from, you might want to pick a different GCamp that matches the time scale of whatever you're hoping to measure out of your neuron. So those are all different things that we can do in terms of picking the right GCamp. What we can do with this technology is pretty amazing, and so I'm going to just walk you through a couple of examples of relatively modern examples of what we can now do with this technology. I talked to you earlier about electrical recordings being able to measure from lots of different cells, but you don't really know where they are relative to each other. And even if you can measure from a lot of them, you can't see the ones that are right next to each other because your electrode arrays can only be so far close together. Even the NeuroPixels probes, each shank has very dense electrodes, but you don't see the ones that are right next to it because the next shank you apply has to be a minimum distance away in brain space. With movies of neurons, however, because now we've turned this into an optical problem, you can actually just use microscopy tricks to image cells that are close together and then change the focal plane of your microscope to see a Z-stack of neurons at different depth. Okay, you can make yourself a little volumetric movie of neural activity. Depending on how fast the neurons are firing and depending on the kinetics of your GCamp, you can scan through a volume of brain to monitor the electrical activity, the neural activity of those neurons in a pretty large volume, depending on how fast everything goes, depending on how fast your lasers are and how fast your optics are, how bright your GCamp is, all of those things. Okay. There's a picture here also that label is imaging through a thin skull. The really cool thing about imaging different types of GCamp activity, different kinds of calcium imaging, is that sometimes you don't even actually have to go and drill a hole inside the skull. Sometimes you do, depending on the access, but sometimes these things are so bright that you can just image it right through the bone. Sometimes when mice are really young, their skulls are really thin, and you don't even have to thin them. You can just image right through the bone. For slightly older animals or animals that have thicker skulls, you don't necessarily have to drill all the way through and remove the bone. You can just kind of thin it. Think of a polishing device. You get a little dental drill and go, you can thin the skull a little bit, and then you can image right through the rest of it. And that's really cool because it makes this technology slightly less invasive than necessarily taking a giant stabby sword and stabbing it into the brain. So it's possible for this to be a slightly less invasive technique than electrical recordings. We could also take advantage of this to do calcium imaging in awake and behaving mice. This is something that is a technology that is possible with electrical recordings as well. We know how to do this where you put a head stage that's implanted inside, that's sticking out of the animal's head, and the animal can either be sitting in a chair and behaving, or they can be running around in an arena and behaving. If they need to be attached with wires, sometimes you have to build a special set of wire management systems so they don't tangle themselves up or chew through the wires. It is now possible to do wireless recordings as well, and there's also wireless microscopes that have been developed, so you can do calcium imaging in awake behaving mice, not head fixed either. You can actually do a microscope that kind of runs around with the mouse or whatever it is. Okay, cool. So that's a lot, but let's just walk through this one experiment first. In this one experiment, what they've done here is they've taken one of those large styrofoam balls that you might go and get at the craft store. It's like, for a mouse, it's about that big spherical styrofoam craft ball, and you float it on an air table, so it's floating on air, and the mouse is head fixed, so it's glued through its head on the bottom of a microscope, so its head can't actually move, but the rest of its body can. So this is what's called an omnidirectional treadmill, of the kind that you may have seen. People have actually built versions of this for humans. It's just gigantic if you have to build one for a human. And it's also proposed as one of the more plausible ways of doing virtual reality for humans as well. You want an omnidirectional treadmill, because you can only run in one direction. It's not particularly interesting. So these mice are running on an omnidirectional treadmill. And then in addition, they have a really cool movie that they can watch that is built as a video game engine. So this is basically a mouse in virtual reality playing a video game. And the projection screen in this particular case, instead of using, sometimes people just use computer screens that are flat. Mice have visual systems that don't just face forward like ours do. They can kind of see significantly farther behind them than we can. It's not exactly 360, but it comes pretty close. And so you have to kind of convince them that they have a credible visual environment by putting sometimes several computer monitors kind of around them. Or in this case, what they've done is a toroidal screen with a projector that projects it onto a movie screen, but the movie screen is curved and goes all the way around the mouse. And that's pretty cool as well. So you convince the mouse that it's actually seeing something like the way that it's expecting to see it, and it can move its head, which does feel a little weird, but it can still run and turn and go forward and left, and it can go backwards if it wants to as well. And because this entire thing is underneath it's a microscope, and you have calcium indicators inside the mouse, you can measure the electrical activity, the neural activity of cells inside the brain as it's doing a navigational task like the following one. So that's the virtual reality system. That's a video game the mouse is playing. Maybe not the most, I mean, it's not Halo. It's not quite exciting. Like, you may not want to play this game. But with a mouse, it's running through a maze in a way that is semi-credible, and it's able to behave in a way that is relevant for the animal, and solving a task that's actually relevant for the animal because it's really good at navigating because it's a rodent, right? So this is a really cool experiment that's enabled by calcium imaging, and you can actually image all the way down into some deeper structures in the brain as well, because it is, tricks were played so that it was optically relatively transparent. So that's imaging down into the brain at pretty good resolution. One trade-off that is possible to have is, instead of imaging at high resolution, you can see every single cell in a small area, if you just put the mouse under a different objective and had it zoomed out, you can measure from the entire cortical surface of the mouse. You can't see single cells, but you can see everything on the top of the brain of this mouse. I'm going to walk you through how that works, okay? So once again, you have a mouse that's glued underneath a microscope objective. The mouse's scalp is completely open now, so you can see the entire dorsal surface, the upper surface, the dorsal surface of the mouse's skull. You thin the skull and you can image right through it. And because the mouse's brain, its cortex, is not bunched up like ours, it's not wrinkly, because it's not that big, right? This is just a coincidence. It was a convenient coincidence. You can't really do this with wrinkly cortices like ours, because there's folds that you wouldn't be able to see. In the case of a mouse, you can actually see the entire thing because it is actually a flat sheet. So you can see the entire dorsal surface of the mouse cortex by imaging with calcium imaging directly down. And so what you're seeing here is, this is a dorsal view, the top-down view of a mouse brain, annotated by the different brain areas. These two bulby things up here are the olfactory bulbs. We're not imaging them, but you can actually image the olfactory bulbs. In this experiment, they're not. And what you get out of it is a movie of delta F over F, the calcium activity that is correlated with neural activity of everything that's going on in the entire brain. Think about what that means. This means that instead of looking at isolated pixels in that movie I've been trying to watch, I'm watching a movie trying to figure out what's going on. Instead of now watching individual isolated pixels, I am now watching something much closer to the entire movie. It might be at a not great resolution, because I can't see every single cell, and I also can't see everything underneath the cortex. So it's not complete, but it's significantly better than what it was before. And furthermore, I'm able to see neighborliness. I can see cells that are closer together, and I can see cells that are farther apart, instead of just having cells that are firing sort of blindly in space and knowing not where they are. So there's lots of trade-offs, like there always are in choosing different types of technology. So I'm trying to point out here the relative merits of electrical recordings and optical recordings. In reality, neuroscientists are very much in a rat race, trying to push the forefront of all of these different technologies. And so I'm really looking forward to what's going to happen in the next couple of years, especially because this type of technology is being developed very, very quickly. And especially the efforts of lots of different collaborations, like the International Brain Laboratory, the Allen Institute for Brain Science, the different collaborations around the world that have been large collaborative efforts are doing these sorts of really large-scale experiments and then just putting the data on the internet. Think about that for a second. Your ability to be able to do neuroscience used to be limited by your ability to grind glass pipettes. If you were really good at grinding a glass pipette, you got really great data. We are starting to enter into an age where neuroscience data in very large, overwhelming quantities are being collected of the highest caliber by the best technical minds and being put on the internet so that anyone with the internet connection and a computer can try to analyze it if they felt like it. We will see what happens. It's going to be a brave new world. Very excited. So being able to record from the entire cortical surface of a mouse is pretty cool, but again, like I said, the cortex is only the top part of the brain. I like the cortex as much as the next person, but we must recognize the cortex is not all there is in the brain. There's a lot more neurons that are underneath. In a mouse, it's not really possible to optically image from all of them. You can put neuropixels probes down there, and people are doing that, but you can't really image all the way down. What you can do is image all the way down in a zebrafish. A zebrafish is a small fish, and a larval zebrafish has the amazing property that it is basically optically transparent. They're really small, and they're transparent, naturally speaking, so we don't have to do anything. And so there's a series of papers and a couple of really cool experiments being led by a couple of different labs where you can now image, using calcium imaging, the activity of basically every single neuron in the zebrafish brain. Just think about that for a second. We have a vertebrate brain. It's not the same as a mammalian brain because it has a couple of things that are relatively different sizes. Fish brains are, they're different than mammalian brains. But in any case, it's a vertebrate brain with a central nervous system and a spine, just like ours. And we have the ability to practically image from every single neuron. This is closer to the picture that we were painting earlier about watching the movie as a whole, right? Now, this may not be the most exciting movie ever. This may not be what you want to do. We want to be able to do this in a human or a mouse or whatever, right? I mean, you might want to do that. I'm not sure I want to do that, but people want to do that, and it's a worthy thing to do. That's not quite possible yet, but it is possible in the zebrafish, and that's pretty cool. Okay, so that is all the things that we can do in terms of optical recordings of neural activity. Just like we talked about with electrical recordings, there is a set of techniques for recording activity. And then kind of on the flip side of that coin is that we also want to be able to manipulate the activity. Now, there's a couple of different reasons we want to do that, and one of them is that being able to manipulate neural activity directly gives us a lot more control experimentally and gives us the ability to ask questions that are not possible to do with passive observation alone. And what we can do in the optical realm is a set of technologies. I got a little bit out of order here. I apologize. I'll get to the thing I'm talking about a little bit. First one slide on voltage sensors. So we talked about calcium imaging, and that's really cool. And there does exist ways to image voltage directly with voltage-sensitive dyes and voltage-sensitive proteins. They're like a similar concept to the calcium imaging, except these are sensitive to voltage changes directly as opposed to changes in calcium. Like we talked about with the calcium sensors were using calcium as a proxy. So you were never actually measuring the membrane potential. You're measuring a proxy of a membrane potential because it is correlated with the influx of calcium. This is a couple of downsides. One, calcium has its own dynamics, right? So you're kind of measuring that, not the neuroactivity itself. And the second downside, and this is a perpetual worry in everyone's head, is that calcium is being used as a second messenger in the cell, which means it has a function, right? When you add a calcium-sensitive dye or a calcium-sensitive protein into that intracellular solution, you're adding an additional target for the calcium to bind to, which means you're a little bit worried about whether or not the cell is still doing what it would have done had your exogenous dye or protein not been in the first place. This is a worry. We try to get rid of it. Depending on how you engineer your proteins, it could be better or worse, right? In the worst case scenario, your attempt to image the cell, you're actually changing a lot about how the cell works, and we don't want that to happen, okay? So there's a parallel set of efforts. People, again, for decades have been working on voltage-sensitive dyes, these small molecules that change conformations in a voltage-sensitive way and can change the fluorescence. And there's also a large set of efforts to build voltage-sensitive proteins. Here I'm just showing you one example that's based on FRET, Forster resonance emission transfer, FRET, Forster resonance I don't know what E is, transfer, T. So FRET technology, with these voltage sensitive proteins, this is not the only approach to it. You can have other categorically different ways of building voltage sensitive proteins that have different trade-offs. Without going into the details, it has the same advantages as the GCAMP do, which is that they can be genetically engineered and targeted so that you can actually put them into specific cell types and have control over where they are and have it be stable inside the cell instead of having to worry about kind of opening up the brain and then sticking in and pipetting your voltage sensitive dye somehow. That's pretty cool. Now, why don't we use these more often? Well, I think the short story is that these voltage-sensitive proteins are just less mature of a technology than calcium-sensitive proteins at the moment. Calcium imaging is super common. People do it a lot. There's a lot of people developing better different versions of it. There's relatively fewer people developing voltage-sensitive proteins right now. This may not be the case in the future. And there's kind of different technical downsides to using these voltage-sensitive proteins that are kind of finicky, and they make them a little more limiting in certain ways than calcium indicators. But nevertheless, they have trade-offs because you're imaging the voltage directly instead of imaging the calcium, and that makes them very attractive. And so I think it's going to be interesting to see in the next several years what's going to happen in the realm of voltage-sensitive proteins as more of them get developed. Okay, so that's calcium sensitive proteins and dyes and voltage sensitive proteins and dyes. That's super cool, right? All of these are optical technologies, so we can hook them to a microscope and image neuroactivity directly. We can also manipulate neuroactivity directly with light. This is a field called optogenetics, and it is one of the coolest things in modern neuroscience ever, because there's lasers. Not just because they're lasers, but there are lasers. So this is a rat that has a fiber optic cable that's piping blue light directly into its brain because you can change its brain activity by shining light into it. You don't actually need a laser. You can actually just do it with an LED. That's totally fine too if you have a bright enough one, but lasers are cool, so we have lasers. Okay, so I'm gonna show you how this works. And this is just a fascinating story that is a rapidly emerging technology in terms of what's been possible now that is pretty much routine. A lot of people do this all around the world in different labs that just, I mean, this technology came out like 10, 15 years ago, and so it's kind of brand new in the grand scheme of things. So we talked about imaging, right? We can measure photons. Here it's the opposite. We're using photons to manipulate neural activity. So this protein here is a protein called channelrhodopsin, and it is a light-sensitive ion channel. I'm going to say that again. It's an ion channel, just like we talked about in all of the previous lectures. We talked about leakage potassium channels establishing the passive and resting potential of the cell. We talked about voltage-sensitive sodium channels and voltage-sensitive potassium channels as the basis of the action potential. We talked about voltage-sensitive calcium channels as being something that's really important for the cell's activity and its maintenance and its second messaging system. In the same way, this is an ion channel. In particular, it is a cation channel. When it opens, sodium and potassium can go where they want to, according to their respective Nernst potentials at the time. But instead of being activated by voltage or being activated like the NMDA channels and the AMPA channels for glutamate, so those are ligand-gated ion channels, this is a light-sensitive ion channel. When you hit it with blue light, it opens and allows cations to flow as they will. Where do we get this protein from? It turns out it is from the green algae. So this is a discovery that crossed the oceans, and it's a protein that's found in green algae, a small single-cellular photosynthetic single-cellular organism, and for them, it's a way of doing phototaxis. This channelrhodopsin allows them to follow the light and kind of go where there's more light so that they can, this is a light-seeking behavior that's done by the single-cellular organism. And so once this protein was discovered in green algae, people started putting it, engineering it, and putting them in animal cells, and then shining them with light and getting them to open. And what you can see here is that just like in the voltage-gated sodium channel case, okay, once you open it, because the Nernst potential, the resting cell, is so far away from the positive Nernst potential of sodium, the net effect is that there's a rapid influx of sodium inside the cell, thus activating it. And so channelrhodopsin, if you manage to stick it into the membrane of an animal cell and you hit it with blue light, what you'll see is that you can make it fire very precisely by shining it with light. Here's a couple of examples. Down here, you can see that every of these orange ticks is one flash of light. Every time it flashes, the cell fires reliably. If you flash them faster, the cell will fire faster. This obviously has a limit. It doesn't go to infinity, but you can see that it's reliably and faithfully following the flashes of light and firing this particular way. So now we have the ability to make cells fire with light. That's good. We can activate it with light. We can make it more active with light. We can also do the opposite with a channel protein called halorodopsin, which is a light-sensitive chloride pump. I'm going to say that one more time. It is not an ion channel in the way this cation channel, the channel rhodopsin is the cation channel. This one is a chloride pump. So it actually uses the electrochemical gradient of protons in the cell in response to yellow light to pump chloride ions inside the cell. Halorodopsin, I told you channel rhodopsin is from the red algae. The halorodopsin is actually from an archaea bacteria. Okay, so another single cellular organism that made this protein. Now we've stuck it into animal cells. And because it pumps chloride into the cell in response to yellow light, it has the ability to silence the cell. So here you see there's a cell here that's firing regularly. Every time you turn the yellow light on, it actually stops firing. Pretty cool, right? Okay. So now with channel rhodopsin and halorodopsin, there's actually a couple of different versions of each of these proteins, and there are also different optogenetic tools that are not channel rhodopsin and halorodopsin, but these are sort of the minimum you need because one allows you to turn something up, the other allows you to turn something down. You now have an unprecedented level of control over the neural activity of whatever neurons you can manage to put these things in reliably. Because these are proteins, just like our calcium indicator GCAMPs were, what you can do is choose to express them in transgenic animals or by viral delivery into non-transgenic animals with promoters that makes it so that they're targeted towards specific cell types. You can put a promoter, say, for an inhibitory GABAergic cell into channel rhodopsin, put it into the mouse, for example, right? So then only the GABAergic cells express channel rhodopsin. If you then deliver the light over the entire cell population, only the GABAergic cells would fire because they're the only ones that have channel rhodopsin. This is really important because in contrast, we were talking about earlier with electrical stimulation. Remember we talked about you're passing current and so all the cells in the area start firing, excitatory, inhibitory, whatever, they all fire because it's electricity. Here, we have cell type specificity. You can deliver the light to all of the neurons, but only the neurons that you've targeted with your channel rhodopsin are receiving that light because the blue light only makes those channels open. So you can then actually dissect the circuit in a way that's completely impossible to do with electrical stimulation alone. This also gets around the problem of exciting fibers of passage, because those fibers of passage, the axons that are just passing by the neighborhood, they don't live here and they're not going there, they're just passing by, they don't have channel rhodopsin either. And so allowing you the possibility of very precisely, with temporal precision, as well as cell type specificity, target specific populations of cells, turning them on or off as you will. That's a lot of control knobs, and so that's why this is one of the coolest things that's been happening in modern neuroscience, and people have been using it to answer all kinds of questions that were just categorically impossible before. So optogenetics has a lot of different strengths, like we talked about. You have precise targeting of neurons to stimulate, and this is true in a lot of different dimensions. If you use an LED, you can bathe a bunch of different brain areas in light, and so you can deliver a lot of photons at the same time. If you target it with lasers and fiber optics, you can target very small areas. And so if your channel rhodopsin is not that specific to a specific brain area, by targeting your laser and a fiber optic cable, you can actually target it to a very small area if you wanted to, thus achieving high spatial precision. You can precisely simulate only the cells in the area that you want to. Channel rhodopsin, as you saw in the previous slide, has very high fast kinetics, very high temporal precision as well. When you shine the blue light, the cell immediately fires the next potential, and when you turn it off, it immediately shuts off. It's super duper fast. And so unlike GCAMP, there's not this lingering effect of things kind of staying open for a while. It has fast kinetics, so you have temporal precision in space. You have high precision in space as well as high precision in time. And because they are genetically encoded proteins, we have the ability to precisely target different cell types. As long as you have a handle on the type of cell it is, and maybe a couple of proteins that are only expressed in that type of cell, you can use those promoters and try to get your optogenetic tools into just those different cell types. There are some limitations, though, because this is not a magic bullet for all of our problems. It is technically challenging sometimes to get the channel rhodopsin and the halorodopsin and the other little opsin proteins to get into the cell. You either have to make a transgenic animal, which is expensive, time-consuming, and sometimes just doesn't work for reasons we don't understand, or you have to develop some kind of viral delivery mechanism to get the channelrhodopsin or the halorhodopsin into your target. Now, the viral delivery, there's a ton of people working on a really exciting progress in modern neuroscience in terms of different viral delivery tools. People have the ability to engineer all different types of proteins, protein complexes that are stuffed inside these viral vectors, and the viruses have an additional property that is really cool because it's something you can't do with transgenics. With viral delivery of channelrhodopsin and optogenetic proteins, you have the ability to target cells that specifically synapse onto other neurons, because these viruses get incorporated into those axons and they can go back up those axons. So you can inject the viruses in the target site. And let's say I want to have the cells over here. These are the cell type A projects a cell area B, right? I inject my viruses with the channelrhodopsin in them at area B. They get sucked up into the axon terminals of these cells, and they travel back up to the cell, infecting only those subpopulations that project to area B, but not anywhere else. Now when I shine a laser at area A, all I'm doing is stimulating the cells that only project to area B. So that's what I get out of viral delivery of these proteins. So it's a limitation, but to some extent, depending on how clever you are in designing an experiment, it could also be a strength as well. Okay, so I talked about it like you just put it in animal cells and it works. Well, the reality is a bit more complicated than that. It's working really well in some modern organisms that are very popular, like it works super well in mice right now, and other organisms as well. There's lingering technical challenges that I frankly don't entirely understand what the challenges are, but there have been lingering technical challenges in using optogenetics in different types of animals, including there's lots and lots of efforts to use them in non-human primates and humans. There's progress coming out on this front every single year and every single month sometimes, but there are some technical challenges making it so that it's not as straightforward as let's just put this in non-human primates and humans, it'll work. I think there's also clinical trials that are in progress right now that are FDA approved for optogenetics in humans, and so we look forward to a lot of exciting progress in that arena. Okay, so that's it about channelrhodopsin as they work, but I wanted to tell you a related story about the history of the people involved in this amazing discovery of channelrhodopsin and optogenetics. And so this technology, if you look it up, if you talk to anyone, the two people whose names come up first in the invention and the innovation of optogenetics are these two individuals here, Carl Dijsselroth and Ed Boyden. Carl, the work was done at Stanford University when both of them were relatively junior researchers, graduate students and postdocs. Right now, Carl is at Stanford and Ed is at MIT. That's where they are currently as faculty, okay? Both of them have received tons of accolades, and they're extremely well known for this amazing discovery innovation of using opsins, light-activated opsins in optogenetics for precision manipulation neuroactivity. But they, as it turns out, in a story that is significantly less well known even to neuroscientists, were questionably not the first people to have done it. There's another neuroscientist, Zhuhua Pan, at Wayne State University in Detroit, who did exactly the same thing, and he did it probably half a year earlier, at least. Same thing. Discovered the same set of proteins that were discovered by other people, these opsins, or people found them in green algae and archaea. There's a big bank of them. And just like Carl and Ed did, Zhuhua stuck them in mammalian cells and were able to show that if you shined light at it, it caused the neurons to fire. So exact same demonstration, but almost nobody, even neuroscientists, know about the story. Why is this? What happened here? The story is both fascinating and extremely complicated, and it folds in a lot of different aspects of science that have to do with the fact that science is a community and a society just like every other aspect of our community. The scientists are human too, like I say, right? And so among the different factors that led to the fact that Carl and Ed essentially got all the credit for having discovered optogenetics and Zhuhua did not. One of the considerations is that Zhuhua was not able to publish his paper, and so his paper was only published, even though he did it earlier, they didn't publish the paper until after Carl and Ed published their paper. And so by the time this paper came out, it wasn't nearly as big of a deal because Carl and Ed did it first. Why did it take this paper so long to get published? That is a good question to ask. The second thing that is almost certainly a big part of the story is the fact that Carl and Ed, when they discovered light-activated opsins and showed that it worked in these cells, they cast it in terms of they had a very big vision for it. They said, this is a tool that's going to change everything in neuroscience. We're going to do systems neuroscience totally differently now because we now have this tool. They cast a really gigantic vision and told everyone what they could do differently because of this technology. Juhua, on the other hand, was a vision scientist, and so he demonstrated optogenetics in the retina, in the visual system. In his paper, even though it demonstrated the same technical feat, the vision was, oh, we're going to cure blindness. We're going to use this in a subpopulation of people who who are visually challenged, and so we can restore vision. So compare the visions here, pun intended. One vision was, we can use this to improve visually impaired individuals. Not everyone works on it. That's super important, right? Nobody would say that that's not important, but it's a relatively smaller vision that's more prescribed than Carl and Ed's vision, which is, we're going to change everything for all of time, right? No one's going to do anything the same again because of this technology. So that's another factor in terms of why some people's names get remembered, other people's names do not. So for further reading, I recommend the article down below. There's a link there. The story is fascinating, and I really like telling these types of stories, like I said earlier in the series, because I like thinking about the process of discovering in neuroscience, the technologies that we had at the time, as well as the human players, and the very real sociological and community efforts that go into which innovations are widely adapted and which ones are not, which individuals get credit for which things and which individuals do not. So that's kind of a thing that we should remember as a community as well, that scientists are humans too. So that was a whirlwind tour. I talked a lot. I probably talked really fast. So I'm going to do a summary here of optical methods in understanding neuroscience. Well, we talked about how electrical activities of neurons can be imaged by using this trick of using fluorescent molecules, so that instead of using antennae to measure electrical activity, you can actually use, you can turn that activity into fluorescence and then use imaging techniques in microscopy to image this instead. You can do this either with small molecules or with genetically engineered proteins. They both have pros and cons in terms of which one you might choose for your particular experiment. We talked especially for a long time about calcium-sensitive indicators because they fluoresce more in the presence of calcium, and that's correlated with increased neuroactivity. And imaging this neuroactivity is a really cool thing because it allows us to see more cells simultaneously with really large fields of view. So instead of tiny fields of view, it's a couple of cells at a time. You can image from a lot of different cells now simultaneously, and that's a pretty interesting way of getting a more holistic view of neural activity. Because they're genetically encoded, we can target specific cell types, and that's a really cool thing because that's something we couldn't do before. And this is one point that I did not actually make earlier. In contrast to electrical recordings, measuring neural activity with optical methods is actually less biased because electrical recordings, we're doing this spike sorting exercise, right? Spike sorting is biased towards cells that fire large action potentials close to your electrodes, and they fire more of them. If you had a cell that just was kind of a quiet type, kind of stayed to himself, didn't talk too much, it wouldn't get picked up by electrical recordings because you wouldn't see those spikes that often. You can't spike sort if it only talks a couple of times. But with optical recordings, you're not biased to only listening to the loudest neurons in the room. You can listen to all of them because they're in your field of view. And so it's technically a little less biased towards listening to the cells that happen to be spiking a lot, to be talking a lot. And then we spend a lot of time talking about optogenetics because that's the flip side of the coin. We can measure neural activity with light, and then we can now have the technology of being able to use light to stimulate activity as well. These are light-activated ion channels that manipulate neuron activity directly with light, delivered either to an LED or a fiber optic cable hooked to a laser, or any of these different ways that we can manipulate light. And so it's the same technology that everyone's been using to manipulate light in other contexts. So kind of borrowed all of those tools from physics and used them to manipulate neurons instead. Okay, so these are really, really cool tools. I just wanted to get the concepts out there. Hopefully I've hopefully I've convinced you that these are really useful tools, and understanding how they work and why you might choose one versus another, for example, in an experimental context, is something that's pretty useful as we start talking about systems neuroscience, how different areas of the brain talk to each other, how different cell types have different functions. And so these are all the different tools that we can use to interrogate those systems and try to tease apart how it is that sensing and action are tied together and give rise to this closed loop view of systems neuroscience as a system of interactions, both between the animal and the external world, as well as the different parts of the nervous system as they work together.